We are on page 49. 49. No, you're not finishing off stuff right now. Just put it away. Finish page 49. Yes. Thank you. So once again, this is video.
5, 6, 7.
Thank you. Good. Once again, this is a video. We're going to be watching it, and then actually, yep, we are going to be watching it, and then we'll go through it together. Make sure you didn't miss anything. Thank you. Today, we're going to hang out with the Amoeba sisters. I'm waiting for everyone to be focused up here. Both DNA and RNA are nucleic acids. Now, does anyone remember what DNA stands for? Deoxyribonucleic acid. And RNA stands for ribonucleic acid. So they are both nucleic acids. And nucleic acids are polymers. What are the monomers that make up nucleic acids? Phosphates are a part of that. Nucleotides. So nucleic acids are made of nucleotides. So DNA is made of nucleotides. RNA is made of nucleotides. What's the circle? Phosphate. What's the pentagon? Sugar. And what is the rectangle? Base. What do you notice that's different about the DNA and the RNA? They both contain 6 nucleotides. The structure. What's different about the DNA and RNA structures? One has both sides and one just only has one strand. So DNA is doubled. It's double-stranded. RNA is single-stranded. That is an enriched word. It means that, do you see how the pentagons are pointing up on one side and down on the other side? That just means they go opposite directions, okay? But you don't need to know that or know that word if you're not interested. So you are going to be responsible for knowing that the sugars are different. But if you don't want to remember the whole word deoxyribose, you can just call it the D sugar. What do you think you can call ribose? The R sugar. Okay, so they are slightly different sugars. And you can always recognize the name of a sugar because it ends in ose. Have you heard of sucrose?
Yeah.
Glucose?
Yeah.
Fructose?
Yeah.
All sugars. Who does uracil take the place of? Thymine, okay? So it's kind of nice because T and U are right beside each other in the alphabet. Yeah, like STU. And so uracil takes the place of thymine, okay? So U takes the place of T. And in fact, we're just going to pause here for a second. Let's find our time code. We're at 3:01-ish. And we're going to go back here because here's our DNA and it codes for our traits. Have you heard the word traits before? Traits are very much like characteristics, but they're a specific biological term. Okay, so genetic traits are things that you get from your genes. So I got short genes, short trait from my mom. My dad is almost 6 feet tall. My mom is 5'2". And sadly, I cannot reach things on the high shelf because my mom's genes are more dominant. Eye color is a trait. Hair color is a trait. And your blood type is a trait. Does anybody in here know their blood type? Do you know yours? Yeah. Anyone else know their blood type? A few of you. Yeah. I thought everybody knew their blood type. I grew up knowing mine, but apparently not everyone does. And it used to be in school, you could find your blood type. Like we actually did blood typing in class, but we're not allowed to do that anymore because you can't have blood around because you could get like contagious diseases from it. So anyway, there we go. Back to 304. 304. Where are we? There we go. What do we have here? We have mRNA. Does anyone remember what the M stands for? Messenger RNA. It's the one that takes the message of the DNA code out from the nucleus and into the cytoplasm. We also have a thing here called tRNA, and we have one that's rRNA. Anyone want to take a guess what that little r stands for in rRNA based on the picture? Ribosomal RNA. Okay, so that is ribosomal RNA. And this little guy here, tRNA, it's kind of nice because it looks like the letter T. Yeah? But it's actually called tRNA because it brings in an amino acid and transfers it onto the growing amino acid chain in the ribosome. So it's called transfer RNA. Ah, what happened? There we go. Sorry about that. And if you take grade 12 biology, you'll get to use one of these little amino acid charts and build yourself a protein. Whoa, what happened? Let's think about these roles here. What type of protein do you think this one is? What's it doing? What does it look like? It's got a little hard hat on. It's building things. And what do we build? We build structures. This would be a structural protein. Here we have a little protein, and it's got this box that it's trying to get out of the cell. This is the cell membrane. Okay, transport proteins. And here we have something that is, I don't know what that one's doing. I think it's supposed to be an enzyme, but I'm not sure. Any guesses here? We got some little germs. What are these proteins doing? Yeah.
Like killing bacteria.
Yeah, they're killing the bacteria. They could be antibodies, things like that. Okay. Okay, and this is on page 56, I think. Nope, 57. So everyone turn to page 57. And we're going to take the little quiz. See if you can guess. If I have 8 DNA nucleotides, how many DNA bases do I have? You can fill in your answer at the top of page 57. How many base pairs do we have if we have 8 DNA nucleotides? Yes. If you can tell me, if I have 8 DNA nucleotides, how many DNA bases do I have? Oh, that's base pairs. How many bases do I have? 8. Yes. And how many base pairs do we have? 4. How do they pair? They have to be, starts with a C, complementary. Yep. Okay, so see if you can fill that in. It's number 2A on page 57, the complementary DNA for that strand. Okay, here we go. Let's see if you got it right. Okay, do you see how the DNA strands have come apart? Right? We've sort of broken the rungs down the middle. The steps of the DNA have been broken. Do you remember what holds them together, these bases together? Yes. Hydrogen bonds. That's enriched thing. So if you don't know that, that's okay. But they are strong enough to hold it together, but weak enough to open up so that we can make that RNA copy. Yeah? So here's our little RNA copy, and the RNA also has to complement the DNA. See if you can fill in the complementary RNA bases, question 2B on page 57. Okay, what are our bases? A goes with, for RNA, it would be U. T goes with A. T goes with A. G goes with C. A goes with U. And C goes with G. Excellent. Okay, so we're not going to explore that because we don't have time, but we are going to go through here and just make sure we've covered all our vocab words and our concepts. So back to page 49. What words are in here that we should be covering? We have trait, 49. Traits, vocab. Characteristics like hair color, height, eye color. There's actually some really interesting traits that we could talk about because they're very, very simple. They only have one gene that controls them. Eye color is complicated. There's a lot of genes involved in that. Hair color, again, it's really complicated. But if your thumb can do this, you have what they call hitchhiker's thumb. If your thumb does not bend, like if you cannot make it go backwards, you don't have that particular gene. This gene is the dominant gene. So that means that I have at least one copy of the hitchhiker's thumb gene. I might have a straight thumb gene, but it gets overpowered by the hitchhiker's thumb gene. In order to have a straight thumb, you need to have 2 straight thumb genes. This is what grade 10 biology is going to be about. It's going to be about like looking at actual traits and how they're inherited. Okay? Your earlobes. Your earlobe, some people's ears go like this. Some people's ears go like this. If you don't know what your ear does, ask your neighbor. So if you have an actual detached earlobe, an earlobe that doesn't go straight into your head, that means that you have at least one copy of the dominant gene. You could have detached earlobe and non-detached earlobe genes, one from mom, one from dad. Or you could have 2 detached earlobe genes. But in order to have the one that is not detached, you have to have 2 copies of the recessive gene. And like I said, that's what grade 10 genetics is going to be about. Other vocab words, obviously, RNA, DNA. Something I should point out, many people are writing RNA and DNA in lowercase letters. They're always uppercase letters. I'm not quite sure why, but they should be uppercase. Tell your partner, what is a gene? Okay. Lots of stuff here. You are responsible for knowing the location of DNA and RNA. You should be able to compare and contrast DNA and RNA. What does it mean if I say compare? What does it mean? What does it mean if I say compare and contrast? What do I expect you to do? You're pointing out the things that are the same and the things that are different. So compare and contrast is something that you have to do in many classes, but very often in science. Compare and contrast means tell me the things that are the same and tell me the things that are different. So you should know the location of DNA and RNA. DNA is in the nucleus. RNA is in the nucleus and in the Cytoplasm, you find it both places. Prokaryotic cells don't have a nucleus, so everything is just in the cytoplasm. You should know that DNA and RNA are nucleic acids. So DNA is a polymer. RNA is also a polymer. What are the monomers? The nucleotides. Remind your partner, what are the 3 things that a nucleotide is made of?
Base.
Sugar plus phosphate plus base, yes. Okay. I don't think you need to know the word biomolecule. Let's just forget about that one. Okay? If you're interested in that kind of thing, I was, because I love molecular biology. It was kind of what I did my degree in. I did biochemistry and molecular biology. Biomolecules are large molecules in biological systems, such as proteins and carbohydrates. So maybe instead of crossing that out, I can make that a little enriched thing. So we have nucleic acids or biomolecules. What are other biomolecules? Proteins, carbohydrates. Okay. Both DNA and RNA are made of nucleotides. So if you're like, what's something that's the same about them? This is something that is the same. Phosphate, sugar, base. Here's something that is different. Single-stranded versus double-stranded. Yes? Yep. Something that is different. D sugar and R sugar. If you want to know the name deoxyribose, that can be enriched. Ribose, that can be enriched, but you should know D sugar and R sugar. So what's the same and what's different? The type of sugar is different. The type of biomolecule is the same. What else is different? T and U. Okay, we're now on page 54. There's only one type of DNA, but there are 3 types of RNA. DNA stays in the nucleus. mRNA is both in the nucleus and the cytoplasm. Different. mRNA is smaller than DNA. The size is different. You should know about mRNA. You should know what its job is. I don't know how excited I am about... Oh, you know what? Most of you forgot on your test. What is this thing here? It's not, this is the nucleolus, and it is what happens in the nucleolus. It makes the ribosomes. And what does it make them out of? rRNA, ribosomal RNA. Transfer RNA, tRNA. Yes. The nucleolus. Oh my gosh, my pen is so weird. It won't let me just write letters normally. There we go. You should know that the tRNA brings amino acids to the ribosome. And I feel like this year I would like to make codon and anticodon. I would like to make those enriched terms. So the 3-letter code on the mRNA is called a codon. Whoa. The anticodon is the matching. Do you notice that the anticodon is complementary to the codon? We have A goes with U goes with A, and G goes with C. And that little code will tell you to pick the correct amino acid. Okay? So an anticodon is the 3-letter code on the tRNA. The anticodon and the codon are complementary. Here's a tricky question for you. Tricky question. If your codon is AUG and your anticodon is UAC, what was the triplet code on the DNA that came from? TAC. It's the same as the anticodon, except for that it's got T's instead of U's. So if we had this whole thing, we would have AUGCCA. The DNA code would have been TAC and then GGT. What would be the next anticodon for this one here, for this codon? It would be GGU. Yep, very nice. Amino acids join together, they make polypeptide chains. You can just say they make proteins. Okay, the proteins are the polymers. Polypeptide chains are the polymers. And we have transport proteins. We have structural proteins. We have enzymes. And we have the immune system proteins, antibodies. And right now you can work on pages 57, 58, 59. Yes, yes. And remember, you have a quiz tomorrow on what?
On this. Oh, wait. Of this.
Yes.
Of 48.
Uh-huh. 40. And 20. Good, thank you. Hello.
I have 28.
28?
40, 30.
Okay, 28. Why can I not? I'll let you do it. Apparently I cannot turn pages. 40. Good. And 48. Perfect, thank you. Hello.
Hi, so can you go back to the slide with the codons again?
Oh yeah, absolutely.
Thank you.
Excellent.
40.
Yep.
48.
Excellent. Oh, excellent. And did you have the other pages as well? Right, the ones 40 and 48. 40 and 48.
No, I.
Okay, so
Because that was a way.
I'll give you a 3 for now. Okay. Yeah. That was Yuna. Yes. You can. I don't know why I'm trying to do it with one hand with a pen in it. So your homework, 57 to 59, your quiz tomorrow is on pages 29 to 48. Okay, so not the new RNA stuff. You can absolutely take a picture of that. So, can you make a cheat sheet for this? Should you make a cheat sheet for this? Yes. Okay, pages 29 through 48. And then on Monday, there's going to be a quiz on RNA versus DNA. Each triplet. Okay. Don't kill yourself. No, I need my Evelyn, did you show me your homework? No, not yet. I didn't think so. Okay.
Said no more. I'm done. So I went up to him. God wearing black. Just a little war.
I would say, you know, that's my information. Also, the way you made it best to read it.
Yeah. I know.
I did not.
No. Oh my God.
You said an ad down.
What, bro? Calling me your son. I'm really mad. Yes, you did, bro.
Miss Spence,
I'm your
28, 40, and 48.
Okay, let me find my time here. There we go. No, here. Good.
40.
40, good. Excellent, thank you. I'm glad you can read that.
Yes. No, Yale. No. Watch, listen. I don't want to listen. No, bro, I can do it. I'm not saying. I love the open outside. I was still that far. Oh, no. Yeah. Open Chinese, the bats.
Hi. Yeah. Yeah.
48.
Good.
I promise I will. So strange. We were still being overall. Only yourself. Is that good? You have to lie. You think it's a ride or die. I would never tell a lie. There you go. Picture. Looks like a pickle.
Very nice. Good. Very nice. Thank you.
DJ. Alex was like, Alex, I don't want to open your highlights. Who does sometimes? It's so cool.
Okay, well, this is
Yeah, why not?
And then no, but like I'm just
Folks, I'll be right back. I'm just gonna make this a long time.
I think you're because you've got no clout, you've got no emotions. Oh, I'm a emotion. Yeah, oh, so I got those. No money. No, I'm saying, I think over here, my ex girl, raise my number. When I'm done, yeah, right, that's. It says has. Why that was wrong here? What? Looking like the dead is over. Fuck. Oh my shit. You know what? My water, your people, no, I'm so sorry. That's what I like what to look like. That's right. That doesn't matter. You can't see. No. You can't. Your people. He said large. What do they do? Ain't that what they do? The passengers, what? They got Jews, you don't know what you think you're the world, but you don't know Jews. No, bro, no, I didn't say all of this. You. I'm gonna say you're heaven. It's I'm in heaven like a big boy. Yeah, wait, I'm in heaven like a big boy. Yeah. God don't do stuff like this. I'm blow your ass. Or a stick. Get in the head. I'm gonna touch you on the head. We can ignore that. 45 have you on the head. Hey, yo, you're like this side right here. Yo, I know that. I have seen my whole when mom is this way. Like, I'll go get it. That's so funny. It's talking about broken. That's the same, right? Broken. It's like a broken connect to get that. It's a forgot college. Your friends want to look out. There's no connection to the other. Ooh, shut up. Like that almost kind of like really loyal to plumbing. More like what if we have slow right away? So it's like slow. No. Oh, this is my way. Is this mine? Is this mine? Is this mine? Wait. I'm talking a lot, all right? Let it be. Make it ground swims. What? What's been 42,000? Okay, it's
What do you want? CD, I like the show. Go right.
Hear me out. Oh, she wasn't right there. She that's what I thought.
Why does it?
Hold on to your house.
Every day I'm thinking, I'm not gonna. Why does it?
Oh, well,
Your blood infected. They look you up. I go. Look, I went to bar stuff. First sport. I gotta know you.
she was not. Dirty pickles, gas.
Don't get
Like a we call a blind jersey. Not Steve. Oh, God.
Okay, that's all.
Yep. Yeah, that would be Why are you looking at I know I am.
Man, I was can't.
No, mine's going to find it up. I don't have stop.
I don't like this. Really? I don't like eating it. But
You burnt them? So they look like soy. You don't know what that means?
Hey, don't say no.
She will come soon, I think. Oh, so is it right here? Like 3-5 minutes. She wondering, wait, she did it first now? Yeah, I guess. And the other companies here. Except today.
This new thing stuff is like. Absolutely. Yeah, that's how we feel fast.
All I can think about is that yesterday. Actually, no, I know why. Oh, like, podcast. Someone don't. I think I did, yeah. I actually studied up. I think it's kind of
This. Oh, that's a pretty big face. Yo, that's the most refreshed face. Yo, there's not a single little 14 on your face. It's like right? No, bro, that's not like how spell. I'm over it. So the Xbox and we'll draw the motor. Go.
Okay, my friends, come get yourselves a molecule. One that you haven't had before. And then figure out what it is.
I upgraded the Smart enough?
Now it's only the hibernation.
It looks like,
No, SP1.
Yes, but not the one, it's not the one.
It's just like
Like most things, I'm straight for the one.
That's one.
It's only like, just.
Oh, yeah. Oh,